Okay, in morning session we discuss about the chapter number 6, that is permanent tissue. In permanent tissue, that is we discuss that is when meristematic tissue grows and takes a specific role, they lost the ability to divide and thus become permanent. The process of taking a permanent shape, permanent size and permanent function is known as the differentiation. By differentiation of meristematic tissue, permanent tissues are formed. For that, we take activity 6.2. So we take the first, take a plant stem and cut into very thin slice or the sections. Now stain the slice with the safranin. Place one neatly cut section on slide and put a drop of the glycerin. Cover with a cover slip and observe under the microscope. Observe the various type of cells and their arrangement. And from that, we have to observe in a microscope and give the some question answer. Like that first one, are all cells similar in structure? So from this figure, we can say that no, all the cells are not similar in structure. They are varied in their shape. They are varied in size and varied in the structure. The next question, how many types of cells can be seen? So about 8 to 10 different types of cells we can be seen in transverse section of the stem. And third question, that is, can we think a reason why there would be so many types of cells? So each type of cells perform different functions and hence there are different type of cells. Cells performing similar functions form a group. The development and growth of plant is dependent on these cells and hence there are different type of cells. Now we discuss the simple permanent tissue. The simple permanent tissue is made up of similar types of cell and hence they look like each other. And first one, that is a Parenchyma. So these are the unspecialized cells with thin cell walls, live cells loosely packed with large intercellular spaces between them. Chlorenchyma is parenchyma with the chlorophyll. Erenchyma is parenchyma with the large air cavities within. Parenchyma forms basic packing tissue. Now we discuss about the functions of the parenchyma. So it supports storage of food, storage of nutrients, storage of water, photosynthesis in case of chlorenchyma, giving buoyancy to the plants and help in floating in case of the Erenchyma. Second one, Skelenchyma. So it is a living, elongated, irregularly thicked at the corners, very little intercellular space, located in leaf stalks below the epidermis. Now we discuss about the functions of the Skelenchyma. It provides the flexibility, helping in easy bending, provides mechanical support to the plant. Okay, so these are all the functions of the Skelenchyma. Now we discuss that is the Skelenchyma. So these are the dead cells. For example, husk of coconut. Walls are thick by the lignin deposition. No internal space inside the cells. Present in stems around the vascular bundle, in the veins of the leaves and in hard seeds covering. Okay, like the coconut fruits. Now function we discuss. It's provide the mechanical support to the plant. Now we discuss that is a 6.3 activity. We take the freshly plucked leaf of the rio. Leaf of the rio is like a leaf of the aloe vera. Okay, it's just like a leaf of the aloe vera. So we take the freshly plucked leaf of the rio plant, stretch and break it by the applying pressure. While breaking it, keep it stretched gently so that some peels project out from the cut. Remove these peels and put it in a petri dish filled with the water and add a few drops of the safranin. Wait for some minutes and then transfer it on a slide. Gently place a cover slip over it and observe under the microscope. So what you have to observe in the microscope? So a peel of the Rio leaf is mounted on a slide and observed under the microscope. It reveals the structure of the leaf. The cells of epidermis and stomata are seen when observed.